To introduce myself, I'm the chair for this session of Flash Talks, and I am Dr. Jassy Drakulic, and I'm a plant pathologist at the Royal Horticultural Society. Now, this session is all to try and explain, promote, share what the plant health undergraduate studentships are, some of the fruits of those research projects that have happened in the past. So we've got some former students and supervisors in the lineup. These studentships are really valuable opportunities for undergraduates in the middle years of their degree, and it's a collaboration between various different funders, including Defra, the British Society for Plant Pathology, the Association of Applied Biologists, SCI Horticulture Group, and the Colgreve Seabrook Foundation, which the MRSB all kind of coordinates, and there's around 12 projects each year. The students, they carry out the project with their host institution, and they also engage in some communication of their results through an online symposium. And I think they also do a poster of their work as well. So they get to present in written form and as an oral presentation too. And it's just a nice taster of research life, as well as getting the supervisors some valuable opportunities to investigate lines of inquiry that they might not have had time to in their ordinary work and funding streams. So we've got a series of speakers on the line, and I think the first of those is Dr. Gareth Thomas from Rothamsted Research. Before I hand over to Gareth, I just want to emphasize that we won't have time to do a question and answer session during this series of talks, but please, if you do have any questions, our speakers would welcome you to get in touch with them. So please do reach out following the session. So Gareth, are you ready?
Yeah, I'm ready. Can you hear me all right?
Yes, I can hear you great.
Great.
Take it away. Thank you.
So I'll just get my laser pointer out as well. Great. Yeah, so I'm going to talk a bit about the work of a student who worked with us on the Royal Society of Biology Summer Studentship, the Plant Health Studentship. And I'll just say, first off, it's one of the best experiences I've had in my academic career, so I'd strongly encourage people to apply for this scheme. And the talk is the role of underground chemical signalling in plant rhizobia pest interactions, and this was done with a really good student at the University of Bristol, Lucia. So just to make clear that all the work I'm going to be presenting today is her work, and I was just sort of supervising the project. So in terms of some background into the work, we focused on a weevil called the pea and bean weevil. And it's a serious pest of fava beans. And you can see in this image here, this is an adult pest which is causing damage to the leaves and cause substantial losses of fava beans and other leguminous plants. And they've got quite an interesting life cycle because the adults, first of all, will mate and feed in the sort of the leaves of the plant. But then the females, once they're sort of carrying eggs, they'll then move down the plant into the soil, and they'll lay their eggs in the soil and produce these larvae. Obviously, with a lot of sort of herbaceous plants, they can sort of form associations with nitrogen-fixing bacteria, which means that the plants can take nitrogen directly from the air into the soil, so that can reduce the amount of fertilizer we use. And here's just an example here of what we might expect to see in sort of root nodules. So the plants contain these little sacs, which we call nodules, and these sacs are filled with bacteria, which sort of help the plant to obtain nitrogen from the atmosphere to reduce our over-reliance on chemical fertilizer. But back to the insects for a minute. So as I say, the adults will sort of feed and mate in the sort of leaf foliage. Then the larvae will actually hatch in the soil. And what's been sort of noted over time is that the larvae seem to specifically target these nodules which contain these bacteria. And this is really how a lot of the crop losses occur. So this could potentially indicate that the larvae can detect chemical signals that are being produced by the nodules below the ground, which helps them to identify the food source and eat the nodules and then cause plant damage. So what we're really wondering is if we can identify chemicals or odors released by these bacteria within the roots as potential attractants for the Cytona lineatus larvae. So ideally, we could sort of potentially set up traps which lure the larvae away from the plants and potentially reduce crop damage. So the two sort of main project aims was to determine the behavioral responses of Cytona lineatus larvae to the Vicia faba rhizosphere, and secondly, to identify odors produced by faba bean roots nodule tissue, and the associated rhizobia bacteria. I won't be talking very much about part one, because obviously for this we needed a lot of larvae. And if you remember last year, the summer was very dry, so we didn't have much luck getting the actual larvae for these tests. So really the focus of the talk is going to be on the bacteria side of things, so these nitrogen-fixing bacteria. So the first thing we wanted to do was identify these odours produced by the rhizobia bacteria. And in order to do this, we obtained sort of 3 individual species of rhizobia, which are all associated with these nodules. So the 3 species are Rhizobium meliloti, Rhizobium leguminosarum, should have practiced that beforehand, and Rhizobium trifolii. We've also included growth media, which has not got any bacteria on it. And the growth media just contains all the nutrients the bacteria need to grow. So the technique we use to identify these odours is called dynamic headspace collections. And essentially what that is a way of collecting the odours into a form that we can analyse. So you can see within this glass chamber here, we've got the bacteria culture, and we can push charcoal purified air over the headspace of this culture. And any of the odours that the bacteria are making can then stick onto this tube here, which contains a powder. And this is where all the odours will stick. And what we can then do is take the tube away from this system, and we can run a solvent through. And that allows us to take the odours from the tube and into sort of chemical extracts. And with this, we can analyse this extract to see which odours the bacteria are making. So this is a technique called gas chromatography. And essentially what it does, it takes mixtures of odours and allows us to separate them so we can see what is actually contained within these odour extracts. And where you can see a triangle on these, what we call tracers, that is where there's a compound or an odour being produced. So at the top one, we've got the growth media, which is just the control. We've got the meliloti, leguminosarum, and the trifolii. And just some of the key things to pick out, these asterisks seem to have moved a bit, but there's a lot of sort of odours that are being produced by the bacteria, which aren't in the control, which is obviously good because it shows the bacteria making them. But there also seems to be differences in the amount of these odours produced by the different species. So that's something that was quite interesting. And we did several replicates of this, and it was all very consistent. And the next thing we wanted to do was identify odours produced by the roots of the fava-bean plants. So in order to do this, we used a sort of slightly modified method where we actually sort of separated the plant roots from the rest of the plants. And again, we confined it into this glass vessel and then used the same principle where we collected the roots smells, the odours that are being produced onto a tube again. And what we can see here is that in the case of the blank, so the control, which doesn't have any plant roots, we can see just a few sort of background contaminant odours. But with the roots, we can see a lot of odours which aren't present in the blank sample. And this is quite an interesting finding because we haven't really looked at this system before. What Lucia shows that this is a good method to take the odours directly from the roots in a more sort of ecologically relevant way. So in terms of the conclusions and future work from the work, we've shown that rhizobia show differences in odour production. We've also shown that the method's been developed to capture root odours from non-nodulated Vicia faba roots. We plan to sort of compare the odours produced with ones that do actually contain these bacteria. And the thing that we probably would have wanted to do first in an ideal world would be the behavioural test to see if the larvae are actually specifically moving towards the smells that are made by the bacteria when they're within the root. And yeah, just to sort of give a bit of a background into the added value we've obtained from the studentship. So as I say, Lucia was a fantastic student and she worked really hard over the summer. She's actually contributed to a publication which has been submitted and she is one of the co-authors. Although for full transparency, this paper did actually just get rejected this morning, but we'll be resubmitting it to another journal. So, you know, her hard work is still sort of being appreciated. She's generated a lot of preliminary data, which we're hoping to put into future grant applications or PhD studentship. So if anybody on the call wants to discuss that further, that would be great. And she's also contributed her work in an article within The Biologist, which is one of the Royal Society of Biology's magazines. So she sort of detailed a bit more of her experience at Rothamsted. And yeah, just want to acknowledge the other Rothamsted supervisors, particularly so Lucia was the student who did all the work. So all the stuff I've presented today was all her work. She's a great student, as well as Joseph Butts and John Caulfield, who were co-supervising from the Rothamsted side of things. I'd also like to acknowledge the Royal Society of Biology for funding the studentship. And I've just provided my email address there at the end. So if anyone wants to contact me, please feel free. So thank you very much.
Thank you, Gareth. That's a really positive story. And I love that you've been supporting the student to continue to contribute to these publications afterwards. And we can all appreciate those the importance of these early research opportunities to develop and give confidence even for the next stage. So thank you, Gareth. Our next speaker, I think, was the participant of a former project. Is that right, Aliya? So it's Aliya Santosa, who is at the University of Cambridge. And I think you're going to share both a bit about your project and about your current work. Is that right? Okay, well, if you're ready to share your slides, please go ahead.
Can you hear me okay?
I can hear you and I can see your screen and it's in presenting.
Good, okay. All right. Hi, everyone. I'm Aliya Santosa. Thank you for inviting me to the talk. I had the honor of receiving RSB Plant Health Undergraduate Studentships with Forest Research back in 2021. Just a little bit about me. I did my undergrad degree in biotechnology from the University of Edinburgh, and I'm currently working as research assistant at the Crop Science Centre, University of Cambridge, under supervision of Jong-Min Choi. Today, I will talk a little bit about the studentship, how did that help my career development, and my current work, which is unraveling the environmental factors that suppress arbuscular mycorrhizal symbiosis. The studentship project I conducted is an analysis of invasive Phytophthora pathogens diversities in plant nurseries and pit-free growing media. So the aim is to investigate the risk posed by Phytophthora spread in plant nurseries in the UK. And I was supervised by Dr. Sarah Green and Deborah Matka from Forest Research. A little bit of introduction. Phytophthora species are microscopic fungus-like organisms. They are causing damage to plant species, agriculture, horticulture, and forestry crops worldwide. Phytophthora thrive in plant nurseries, particularly in water, soil, roots, and shaded plants. There were also concerns raised about pit-free growing media, which often contains bark, green waste, and coconut coir in spreading Phytophthora pathogens. Water sources and plant roots were collected from 2 plant nurseries in the UK, as well as 4 sources of pit-free growing media. So the Phytophthora diversity was then examined, which involves first leaves of various bait plants floated on the surface of the water samples and growing media. And then next, Phytophthora isolation were attempted from any developing lesions. Then the Phytophthora species were then identified by morphological methods and quantified using nested PCR, QRT-PCR, then sent for sequencing. So data on these findings was analyzed in relation to host species and nursery management practice. The results when I did the studentship, Phytophthora chlamydospora and multiflora were found from both nurseries. So based on the 101 samples analyzed so far from these nurseries, 57% tested positive for the presence of Phytophthora, and no Phytophthora were identified from pit-free growing media. So the implications of the studentship is a good practice of biosecurity is recommended for plant nurseries to ensure the staff are aware to prevent or minimize the pathogens. And also the studentship also support Defra to detect and control pest pathogens and the risk of plant health. So how did the studentship help my career development? Having the opportunity to join the studentship was a wonderful experience. So the experience increased my understanding of plant disease detection and diagnosis. And I've gained practical training in laboratory techniques, experimental design, and troubleshooting in the lab. I've improved my confidence and also inspired by researchers and students who care for plant health research. And this overall experience has given me the courage and skill sets to perform my current job as a research assistant. So my current work now is trying to understand how environmental factors influence plant root symbiosis with arbuscular mycorrhizal, which I will refer to as AM fungi. So the experimental system involves using Rhizophagous irregularis as AM fungi and rice. So arbuscular mycorrhizae symbiosis is a mutually beneficial interaction formed between most land plants, like around 80%, and soil fungi. So during symbiosis, fungi and plants exchange nutrients. Plants supply the fungi with carbon in return for receiving carbon. The fungi deliver minerals such as phosphate and nitrogen, and then transfer it to a highly branched tree-like fungal structures called arbuscules inside the roots. So besides nutritional value, AM symbiosis can improve disease resistance as well. Protective effects of AM symbiosis against pathogens, pests, and parasitic plants have been described for many plant species. And a paper published from Azel et al. 2019 demonstrate the disease symptoms on tomato leaves infected with tomato mosaic virus. So this in response to mycorrhizal colonization. The leaves of infected tomato plants looks healthier when inoculated with AM than without AM. So AM symbiosis activates plant defense response to allow efficient defense mechanisms against the pathogen attacks. However, the application of AM symbiosis into modern agriculture is challenging due to environmental factors that suppress the symbiosis, such as high fertilizers, cold temperatures, and waterlogging. Therefore, we are trying to understand the molecular mechanisms underpinning the suppression of AM symbiosis under diverse conditions. These are the three treatments plus control that I'm exploring in the lab. And the method involves RNA sequencing to investigate molecular mechanisms behind each of the treatments and see what's overlapping between the treatments. Here's a picture of wild-type rice with and without AM. It looks like AM promotes faster growth and flowering in the rice than without AM. And the graph here shows total intraradical hyphae and arbuscules on x-axis and the colonization level on y-axis. So three environmental factors suppress levels of hyphae and arbuscules in wild-type rice when comparing these conditions to symbiosis promoting condition, which is a low phosphate treatment. It shows that three environmental factors suppress AM colonization levels. So these samples will then be processed for RNA-seq analysis. So the long-term goal is to stabilize AM symbiosis in diverse environmental conditions to make plants more resilient to pathogen attacks. There are ways to support me and my current work. First, collaboration in sharing genetic resources, displaying altered response to temperature and water treatments. And second, a small research grant to perform an RNA-seq experiment to study global transcriptome changes under various environmental conditions, and an MPhil or a PhD opportunity. Please feel free to reach out through my email if you have any questions. Lastly, I would like to thank everyone from Joy Lab, Sarah and Debbie, also SCI Horticulture Group, RSB for sponsoring and organizing the Studentship. Thank you very much, everyone.
Thank you, Aliya. What a lovely presentation, and spanning quite a lot of different approaches used and different skills that you picked up and transferred from your project into your current work. So all the best for your future opportunities. And yeah, I think highlighting the funding issue as well, like so, because a lot of these approaches to try and find out these mechanisms do cost a lot of money. And that's another benefit of these projects is it's just for inexperienced or early career supervisors, opportunities to put small applications together and build your confidence in applying for funding and writing out your ideas in a formal way. It's another nice training opportunity that the supervisors also benefit from. So thank you, Aliya. Our next speaker is Sebastian. Dr. Sebastian, I hope I say this right, Evers van der Akker. Are you there?
Yes, I'm here.
Great. So you're also at the University of Cambridge. And yeah, if you'd like to start sharing your slides, please.
Sure. Can you see this okay?
I can see it. I can hear you. Go for it.
Perfect, thanks. So yeah, thanks for asking me to talk. I'm a big fan of the RSB Plant Health Summer Studentship scheme. We've had 2 students from this scheme. You can see them. One is here in the mirror and one is Beth here. Both of these students were really fab and went on to PhD. So Nimira is in, I think, the Gurdon Institute in Cambridge, and Beth is with us. So today what I thought I'd do is just tell you about a bit of the work, the kinds of things that we do in the lab, because of course, you know, otherwise I would have to pick one of those 2 stories. And I've entitled it The Maker's Solution to the Biologist's Problem. So we are biologists, but some of us in the lab are also makers. So as in, like, in our free time, we like to make things. And this project is the most fun project I've had in the lab for a long time, because it kind of combines our hobbies with our work. So in the plant parasite interactions lab, we work on plant parasitic nematodes primarily. And I'm not going to go into the detail of this, but they're a big problem basically all over the world. And they're a bit of an overlooked problem. And the reason for that is because they're really hard to work with. And so one of the big bottlenecks in working with plant parasitic nematodes, and this is going to sound trivial, but it's true, is answering these kind of questions like how many nematodes are on the plant, and you know, what is the phenotype of the nematode on a given plant. And this problem stems from the fact that they are below ground pathogens that infect the roots. And so we decided we want to try and solve this problem. And so what we did was, this video is not playing. What we decided to do is to basically buy a 3D printer and then design and print some machines that would allow us to rapidly image infected plants. So you can see that happening in these videos where you've got petri dishes with individual plants, one per petri dish, that are being processed by the machine. And these have been infected with nematodes. These videos are real time, and so one of these machines will do 1 plant in 6 seconds. So we printed 6 machines, we get 1 plant per second, so we get 10,000 plants in 3 hours. This is a picture of what this looks like. So imagine you're under a plant and you're looking up at the root system, and this is what you see in this case. And these white dots, each one of the white dots that I hope you can see, is the nematode infecting the plant. And then what we did was we gave these images to the AI team, so Jisoo's lab, and he was able to invent software that could then recognize the nematodes on those plants. And you can see a kind of a markup of that on the left-hand side and the accuracy of that on the right-hand side, so extremely accurate. And this nematode recognition software was not just able to find the nematodes, but also find out things about the nematodes that are really important for agriculture. For example, the size of every nematode, the color, the shape, the roundness, etc. And in addition to all these important phenotypes, the other phenotype that it gives you, which doesn't sound important, but actually it's the most important, is the position on the plate. So where on the plant is the nematode? And this is a really important phenotype because this kind of phenotyping is non-destructive. And so if we know the position, we could image the same plant the next day, and then we can, you know, join up the same nematode over time. And in so doing, we could assess the dynamics of these phenotypes, or the dynamics of pathology. And so this slide is showing you an example of that, where we've got a couple of images of one plate over time, and you can see we've lined these up, and you can, I hope, appreciate how you can see the plant grow, but also you can see the nematodes grow. And so we can take these images and interpolate a video that hopefully should start playing on the bottom, and you can watch these nematodes grow, and then you can watch them lay eggs. So just lay eggs, the eggs will hatch, and then the next generation of the nematodes will then go and infect other parts of the plant. So this is what we call holistic and dynamic pathology, i.e. we can see the whole plant, all the individual parasites, and track them for their entire life cycle and beyond. And this kind of capability is, really amazing for us in the field, at least. It allows us to look at a whole bunch of phenotypes that are important for the field, and yet we've never had the ability to look at them before. So we decided to use this new capability in the biggest nematode infection trial in the combined history of the human race. And so that's what you can see here. So here you've got 5,000 plants on this bench, 5,000 on the other bench, so 10,000 plants. It's actually 550 genotypes, each with 20 biological replicates. And then we moved the imaging machines into the greenhouse. So you can see this 3 pairs of imaging machines. And then every other day for 90 days, 4 people from the lab went into the greenhouse and imaged all 10,000 plants using the machines. And so over, the period of April to June last year, we phenotyped something like 400,000 events. And then each one of those events, each plant, of course, has got lots of nematodes. So it's tens of millions of nematodes. And this is more nematodes than have ever been measured in all previous publications combined. So it's a lot of nematodes, and yet, of course, it's not just, it's not really actually 400,000 images. It's actually more like 10,000 videos, each with 40 frames over a 90-day period. And so what we've been doing now is extracting the phenotype of the nematode from these videos effectively, and then answering biological questions with this information. I'll tell you about, just touch on 2 examples of the kinds of information we can extract from this huge data set. One is just a fundamental discovery, so just trying to understand more about the nature of the parasite itself. And then the second one is trying to be a little bit useful with this capability. So the first one, answering a fundamental question. So the question is basically this. So if there are more than one nematode infecting a plant, and 2 of them are very close, so infecting the same part of the root, will those 2 parasites help one another, or will they hurt one another? And to answer this question, we could basically just go into the image data set, find every nematode on every plant, so this is a nematode, and then we just draw a circle around it, and then we keep enlarging the circle effectively until it touches another nematode, and at that point we stop. We say, okay, that is the personal space of that given individual, and we plot that on the x-axis, and on the y-axis we plot the size of that individual. And we do that for all the nematodes, so there's some 60,000 biological replicates. And what you can see is 2 things. One, there's basically no interaction over a long distance. So whether a nematode is 1,000, the nearest nematode to me is 1,000 pixels away, which is like a half a plant, or 100 pixels away, I am the same size. But if there's another nematode really close to me, then I'm smaller. You get this big skew to the low end. And so our interpretation of this is that nematodes that infect the same part of the root compete with one another. So that was really cool for us to learn something new about the nature of a parasite that I've worked on, at least for my entire career, and yet we didn't know this. And then the second thing is to try to be a little bit useful. So it turns out these, this wasn't a random set of plants. We picked a sequenced mapping population. So i.e. all of those lines were genetically different, and we knew those genetic differences. And so then what we could do is correlate the genotype, so the genes of each plant, with the phenotype, how many nematodes, for example, and identify parts of the plant genome that contribute to that nematode phenotype. And I won't explain this in much detail other than to say, here are the 5 chromosomes of the plant at the bottom of the screen, and where you can see the black dots, those are the parts of the plant genome that strongly correlate with, in this case, the number of nematodes on that plant, so i.e. new resistance genes. You can imagine there's a lot more we can do with this. That just gives you a flavor of the kinds of things we do, trying to understand fundamental questions in host-parasite interactions and trying to deliver something useful, i.e. new resistance genes in agriculture. I have to thank everybody that took part in this, and of course, the variety of funders and you for your attention.
Thank you very much, Sebastian. What an incredible project with just some absolutely mind-boggling numbers. And yeah, just a really nice creative project, as you say, bringing together skill sets from inside and outside of science and plant pathology. And I can only imagine how inspiring that would be for the students that were in that lab. And I'm not surprised they're still inspired and working hard at PhDs now. Okay, so I think we have one more speaker for the Plant Health Undergraduate Studentships, which is another former supervisor, Dr. Sarah Green at Forest Research. Sarah, are you there?
Yeah, no, thanks very much. And Aliya's already sort of spoken a little bit about the project. And yeah, we were very fortunate to have Aliya for the summer in 2021. Of course, it was just post-COVID, but we had a couple of other summer students as well doing projects. So it was quite a nice, lively summer after a year of kind of being very quiet. So yeah, as Aliya said, the project that she was working on was looking at Phytophthora in nurseries and peat-free growing media. Next slide, please. And apologies for those who've maybe seen this slide before. It's my general introduction to Phytophthora, which I thought might be quite useful to explain why we're interested in Phytophthora, in particular in nurseries. So Phytophthora is Greek, it means plant destroyer. It's a large genus of pathogens. They're oomycetes, they're not true fungi. That's largely because they spread via free-swimming zoospores. In the top right photograph shows a sporangium, which is a spore sac in water releasing these zoospores which can swim. And the swimming ability of zoospores is very important for enabling their spread, particularly in nurseries. And also they're born in soil and water, and you can see this thick-walled resting spore in the middle, can persist for long periods of time in plant debris and in soil. And again, it's this feature of the life cycle of Phytophthora that make them so well able to thrive in the nursery environment. Mostly Phytophthora infect through the roots, but some species infect through aerially. And you may have heard of Phytophthora ramorum, which is killing our plantation larch across the UK. We've got over 180 species of Phytophthora described worldwide on a very broad range of hosts. But the problem is over the last 20 to 30 years, we've been dealing with a number of invasive Phytophthora species that have emerged from sort of unknown locations and have spread largely probably in the plant trade. We have a lot of evidence to show that these Phytophthora are spread in traded plants, particularly containerized plants, and then they're able to spread out into the wider environment. And we're dealing at the moment, as a forest pathologist and forest researcher, and been dealing with a lot of different invasive Phytophthora, not only killing plantation species, but also killing our native species as well. For example, Phytophthora ostracidri killing juniper. And next slide, please. So the project that Aliya worked on is actually an international project. It's a UFresco project, and there were 9 different partner countries involved in the project, and 6 of the countries we were all doing the same nursery sampling methods that Aliya described a little bit. So this is a photo here shows Aliya with a couple of colleagues from the James Hutton Institute who we collaborated with on the project at one of the plant nurseries that we sampled in the summer of 2001. So the other countries that were also involved in sampling were Italy, Greece, USA, France and Ireland. So it was great because we were able to share our methods that we have developed at Forest Research and at the James Hutton Institute and share them across countries and try and engage with nursery managers, engage with stakeholders in the trade to try and raise awareness of the spread of Phytophthora in trade. So water samples, as Aliya said, were collected and root samples were collected as well for analysis. And then 2 different methods of analysis. There was dating that Aliya described in the photograph below, but also we used an eDNA metabarcoding methods. And the results of that came after Aliya had left the project. It takes quite a while to build up enough samples to actually run a plate. So I can present on some of those results today. And a lot of those methods were developed at the James Hutton Institute. So next slide, please. And I'll actually run through some results because I thought maybe you'd be interested in that. So we had 2 nurseries. They're both producers of hardy nursery stock, relatively large. And what's quite interesting, one of the reasons why we selected them, because although they have a mix of home propagated stock, they also import quite a lot of stock from various European countries, but also from outside of the EU as well, New Zealand, Ethiopia and Kenya in this case. Mostly supply the landscaping and amenity sector, so they're not forest nurseries. And each nursery was sampled twice. And again, here's Aliya and Debbie and myself and one of the nurseries. Next slide, please. Next slide, please. Yeah, so just quickly then some results. So Aliya mentioned some results from the baiting that she did, where I think it was Phytophthora gonopodioides and multivora were isolated, was that right? But also the metabarcoding enables us to identify a much greater range of species. So this nursery, we found 26 known Phytophthora species across the 2 sampling visits, quite a high diversity. Of interest, we found Phytophthora ramorum on rhododendron. This is a quarantine regulated pathogen. It's the one that's killing our larch and really writing off plantation larch across the UK. And the top left-hand photograph shows Phytophthora ramorum on rhododendron infected leaves here being grown at this nursery. On the top right, we found Phytophthora ostracedri. This is another invasive species, which is killing our native junipers in the north of the UK, widespread in the wider environment. And this had been imported in the foliage lesions on these twisted Leylandii in the top right photograph are actually infections due to Phytophthora ostracedri. And none of these infections had actually been noticed by the nursery staff. So there was an element there of awareness raising that we need to do. We also found the Phytophthora uniformis, which is one of the parent species of an killing alder hybrid Phytophthora in the roots of prunus and tsuga in a plant dump. And then we found some DNA of a species that's never been reported before in the UK at this nursery. Next slide, please. And nursery 2 had also a very high diversity of Phytophthora across the 2 sampling visits. And again, we've got this Phytophthora uniformis, this parent species of an alder killing hybrid. And we found that on lavender and Physocarpus, which is 9-bark. And then we found another rare Phytophthora also on 9-bark. And then the lower photograph shows box, which was being infected with Phytophthora occultans, which is again a rare pathogen that is spread in the trade. So next slide, please. Aliya did the baiting analysis on our peat-free because we had some concerns about some of the components of peat-free media potentially coming from sources which could be contaminated with Phytophthora. None of the species were baited, but we subsequently went on in a few months after Aliya had completed the project and did the metabarcoding analysis and actually found some DNA of Phytophthora in some of the samples. So just wanted to illustrate that. And that has led to another project funded on looking at further samples of peat-free media. So next slide, please. So I think this is my last slide, and I really wanted to say how much this project was able, particularly its collaboration with a lot of international partners, we were able to work together to build the similar results from across nurseries, all the other nurseries as well. And one of the big factors, which I didn't mention yet, was Phytophthora contaminating water supplies. And our nursery number 1 had Phytophthora in the irrigation water in a reservoir, open reservoir that they used to water their plants, had a lot of Phytophthora. And that nursery manager has subsequently sunk a borehole and is now extracting deep groundwater. So, you know, sort of practices have changed. But what we did do as part of this project was to produce a best practice flyer. It's a 2-sided flyer, and I've got the English version here, and it's being translated into all the partner country languages as well and be disseminated across all the partners in the project. So I just wanted to say we're really grateful to be involved in the studentship, to have Aliya working on this project really helped us along when we were doing the sampling and all the sample analysis. So thank you and well done to Aliya, and I'm quite delighted that Aliya's gone on and found a job also in sort of the area of plant health. And we also have some summer studentship coming up this year as well, working on Scots pine. So thanks very much to everybody. That's it.
Thank you so much, Sarah. And again, it's really nice to see that direct application of the work that we can do as plant health scientists. And yeah, getting that evidence base to support industry to be healthier and support our plants so that we're not just spreading these problems around.